There is something the brain does when visual input disappears that neuroscience manuals describe with precision, but that almost no one outside research laboratories knows about. It is not relaxation. It is not simply the absence of stimulus. It is an active process of reorganization that begins in the first few minutes of darkness and has measurable consequences on the brain's processing architecture. Light is the human nervous system's dominant sensory input. Approximately one-third of the cerebral cortex is dedicated in some way to visual processing. The primary visual pathway traveling from the retina through the optic nerve, the lateral geniculate nucleus of the thalamus, and to the primary visual cortex in the occipital lobe continuously processes during wakefulness a torrent of information the brain uses to construct the representation of the world we call perception. When that torrent is interrupted, when light ceases completely and continuously, the system dedicated to processing it doesn't simply turn off. It begins doing something else. That something else has a name in neuroscience: cross-modal plasticity. It is the phenomenon by which, when a cortical area stops receiving its usual input, it begins to respond to inputs from other sensory modalities. The most documented example is that of people blind from birth or who lost their vision early in life. Their visual cortex actively responds to tactile and auditory stimuli with a robustness not observed in people with normal vision. The cortex, in the absence of its dominant input, repurposes itself. It doesn't wait. It finds another use for its processing capacity. What is particularly disruptive, and what Penrose would find directly relevant to his arguments about the nature of cognitive processing, is that this repurposing does not require months or years of visual deprivation. Studies by Alvaro Pascual León and his colleagues at Harvard Medical School, using transcranial magnetic stimulation to evaluate visual cortex function in volunteers blindfolded for periods between 48 and 100 hours, showed that the cortical response to tactile stimuli in the visual cortex began to be detectable in less than 2 days, progressively increasing with the duration of deprivation. Cross-modal plasticity is faster than previously thought, and its onset is on timescales of hours, not weeks. This has implications extending beyond what happens in individuals with prolonged visual deprivation. It suggests the visual cortex, the largest area of the cerebral cortex in terms of dedicated surface area, is not a functionally rigid region that can only process one type of input. It is a high-capacity processor with considerable functional flexibility, and that flexibility is activated much faster than classical neuroscience assumed. The reason for this speed lies in something Pascual León's studies helped clarify. It is not that the visual cortex builds new anatomical connections in a matter of hours, which is impossible on those timescales. It is that existing connections between the visual cortex and the somatosensory and auditory areas, which under normal vision conditions function suppressed or inhibited by the dominant visual input, become disinhibited when that input ceases. The connection pathways were there. The darkness simply stops suppressing them. In terms of neural circuits, darkness builds nothing new. It reveals what was already present but silenced by the dominance of the active visual system. Penrose would find this directly relevant. The capacity emerging in the dark is not a new capacity. It is an existing capacity the dominant external input was preventing from operating, and that observation extends beyond the sensory to the cognitive, making 10 minutes of darkness more interesting than a sensory deprivation exercise. For shorter periods, on the order of the 10 minutes of complete darkness that interest us here, the mechanism is not massive repurposing of the visual cortex. It is something more subtle and more immediate. the increase in spontaneous activity of the visual cortex in the absence of external input, and the compensatory increase in the sensitivity of the other sensory modalities the brain uses to orient itself in the environment. That increase in spontaneous activity has an electrical signature that electroencephalograms can detect. There is an observation about scotopic adaptation that should be introduced here because it adds a precise temporal dimension to everything we are describing. Scotopic adaptation is the process by which the eyes increase their sensitivity in low-light conditions, moving from the predominant use of cones, which require good lighting and are responsible for color vision, to the predominant use of rods, which are much more sensitive to light and are responsible for night vision. Rods contain a protein called rhodopsin, which bleaches when it absorbs photons and takes several minutes to regenerate. Under full illumination, rhodopsin is largely bleached, and the rods are relatively insensitive. When illumination ceases, rhodopsin begins to regenerate. Complete scotopic adaptation, where rod sensitivity reaches its maximum, requires approximately 30 to 40 minutes in complete darkness. But significant improvements in sensitivity begin to occur within the first 5 to 10 minutes. This means that during the 10 minutes of darkness that interest us, an active biochemical process is occurring in the retina. The regeneration of rhodopsin, the sensitization of the rods, the preparation of the visual system to detect light levels that would be invisible to an eye just exposed to bright illumination. The eye in the dark is not resting. It is recalibrating its sensitivity threshold. It is preparing to see what it could not see before. When visual input is reduced and the eyes are closed in a dark environment, alpha oscillations in the occipital cortex, which have a frequency between 8 and 12 cycles per second, and which, under ordinary conditions, appear when visual processing activity is suppressed, increase in amplitude. These alpha oscillations are not noise. They are the resting rhythm of the visual system, the pattern the visual cortex adopts when it is not processing external information and which is associated with states of internal processing, of thought not directed at a specific external task. It was looking for answers to this that I found this digital book. The Penrose dossier, What Academia Fears to Admit, is not a book about sleep neuroscience or circadian rhythms. It is a tool to develop the disposition, making it possible to take seriously the question of what kind of cognitive processing occurs when the dominant sensory input is suppressed. Penrose spent years arguing that human thought has a dimension that cannot be reduced to the algorithmic processing of external information. Darkness, in that sense, is not just the absence of light. It is the condition under which that non-algorithmic processing can operate with less interference from external input. This book documents precisely that disposition. You are in the precise state where this idea can do something real, not after you have filed it away as interesting. Now, while it is still active, the link is in the first pinned comment, but beware. This content may be taken down. Click now. Download your copy. When was the last time you were in complete darkness for more than a minute, with no phone, no screens, no light source of any kind? What happened inside you? Leave your answer in the comments. But the visual cortex is not the only brain system responding immediately to darkness. There is a system older in evolutionary terms, deeper in the brain's architecture, using the presence or absence of light as its most fundamental calibration signal. That system is the circadian biological clock. The suprachiasmatic nucleus, a small structure of about 20,000 neurons located in the hypothalamus, just above the optic chiasm where the optic nerves from both eyes cross, receives direct information about the amount of light entering the eyes through a specific pathway bypassing the ordinary visual system. That pathway uses a special type of retinal cell, the intrinsically photosensitive ganglion cells containing a photopigment called melanopsin, to send signals about ambient light intensity directly to the suprachiasmatic nucleus, regardless of whether the rods and cones of the visual system are active. The suprachiasmatic nucleus uses that information to synchronize the internal biological clock with the environment's light-dark cycle. And one of its most important outputs when it detects a sustained reduction in light intensity is to stimulate the pineal gland to synthesize and release melatonin. Melatonin is a simple, structured molecule derived from the amino acid tryptophan via serotonin acting as the chemical signal of darkening. When melatonin levels rise, a cascade of physiological changes initiates. Body temperature begins dropping slightly. Blood pressure decreases. Sympathetic nervous system activity is reduced. The organism receives the signal that it is night, that the active cycle has ended, that it is time to initiate the maintenance and consolidation processes the nervous system performs preferentially in the absence of intense external activity. The pioneering work of Richard Wortman at the Massachusetts Institute of Technology, beginning in the 1960s and decisively helping establish melatonin's physiology, demonstrated that light suppression of melatonin synthesis is extraordinarily sensitive. Even relatively low illumination levels, on the order of 100 lux, such as those produced by a well-lit room with artificial light, can completely suppress the melatonin synthesis that should occur during nighttime hours. And the restoration of that synthesis, when darkness is reestablished, begins in minutes. The link is in the first pinned comment. The last units are running out. Click now and start your journey toward the clarity and genius you deserve. The precision of the mechanism by which light regulates the circadian clock was the subject of the 2017 Nobel Prize in Physiology or Medicine, awarded to Jeffrey Hall, Michael Rosbash, and Michael Young for their discovery of the molecular mechanisms controlling the circadian rhythm. What these researchers unraveled is one of the most elegant molecular feedback mechanisms known in biology. The molecular circadian clock operates via a negative feedback loop involving a set of genes whose protein products regulate their own expression. The clock and BMAL1 genes encode proteins that bind and activate the transcription of the period and cryptochrome genes. The period and cryptochrome proteins accumulate in the cytoplasm, form complexes with each other, enter the nucleus, and block the activity of the clock BMAL1 complexes, thereby inhibiting their own production. As the period and cryptochrome proteins are degraded, the inhibition ceases and the cycle begins again. This cycle has an endogenous duration of approximately 24 hours, slightly different in each individual, and is adjusted daily by the light signal the suprachiasmatic nucleus receives via retinal melanopsin. Penrose would have pointed out there is something remarkable in the elegance of that mechanism. A molecular oscillator requiring no external signal to maintain its periodicity, generating its rhythm through the dynamics of its own internal interactions, and simply synchronizing with the environment through a minimal information signal. The periodicity does not come from the outside. It is in the structure of the system. Light does not impose the rhythm. It only calibrates it. There is a detail about the suprachiasmatic nucleus's architecture that is particularly elegant from the perspective of the physics of complex systems. Each of the approximately 20,000 neurons making up the suprachiasmatic nucleus has its own individual molecular clock, with a periodicity slightly different from its neighbors. However, the nucleus functions as a synchronized oscillator, with all neurons maintaining a coherent phase. That synchronization is maintained through the coupling between adjacent neurons via neurotransmitters and signaling peptides. The result is a network oscillator, more robust than any of its individual components, capable of maintaining its periodicity even when some neurons are damaged or eliminated. It is exactly the kind of structure physicists of complex systems describe as a network oscillator, a collective organization emerging from the interaction between components and having properties none of the components possesses in isolation. The biological clock is not in any individual neuron. It is in the dynamics of the network. Penrose would point out the brain is full of such structures, organizations emerging from the dynamic interaction between components and having global properties that cannot be derived from analyzing the components in isolation. And darkness, by modifying the input the suprachiasmatic nucleus receives, does not modify an individual neuron. It modifies the state of the entire network, and through it the state of all the systems the circadian clock regulates. That has direct relevance to what happens when you are in the dark for 10 minutes. You are not interrupting the circadian clock. You are giving it the correct calibration signal, the signal that light has decreased, that the active cycle is ending, that maintenance processes can begin. And in the context of contemporary life, where exposure to high-intensity artificial light during nighttime hours is practically universal, that calibration signal is something many nervous systems do not receive with sufficient regularity. There is a third process occurring in the dark that has a dimension connecting directly to Penrose's arguments about the nature of thought. When visual input, which is the most dominant and interruptive of all sensory inputs under ordinary conditions, ceases, the attentional resources dedicated to processing it become available for other processes. And there is evidence those processes include forms of cognitive activity qualitatively distinct from thought directed at an external task. EEG studies in dark and quiet conditions show, in addition to the expected increase in alpha waves in the occipital cortex, an increase in the coherence of theta oscillations in frontal and hippocampal regions. Theta oscillations, with frequencies between 4 and 8 cycles per second, are the rhythm neuroscientists associate with memory consolidation, processing relationships between events, and navigating complex conceptual spaces. The hippocampus, the brain structure most directly involved in forming new memories and integrating information from different sources, shows increased theta activity under conditions of low external stimulation. This means darkness doesn't just calm the nervous system in the vague sense of the word. It specifically activates the processing mode the brain uses to integrate, relate, and consolidate information. The thought occurring in the dark is not the directed thought occurring in front of a screen or in a conversation. It is the thought that connects, that finds relationships, that reorganizes what is already known in ways the continuous flow of external input normally prevents. The link is in the first pinned comment. The last units are running out. Click now and start your journey toward the clarity and genius you deserve. Penrose would connect this to something he has consistently argued, that the type of cognitive processing producing genuine mathematical insight, recognizing structures transcending any rule system, does not occur in the directed processing mode dominating ordinary wakefulness. It occurs precisely in those states where external processing is suppressed and internal activity can operate more freely. Mathematicians have for centuries described the experience of solving difficult problems during sleep, or in the transition moments between sleep and wakefulness, or during aimless walks, or in the dark before sleep. Henri Poincaré, the French mathematician whose work on non-Euclidean geometry profoundly influenced Einstein, precisely described the process by which the solution to a problem that had occupied weeks of conscious work came to him suddenly as he boarded an omnibus at a moment when his mind was not actively working on the problem. That account, documented in Poincaré's essay titled Mathematical Invention, published in 1908 in the Annuaire de l'Institut Général Psychologique, has become one of the foundational texts in the psychology of insight. Poincaré's account of the discovery of Fuchsian functions deserves more detailed elaboration, because Penrose considered it one of the most illuminating testimonies on the nature of mathematical thought. Poincaré described how he had worked for 15 days to prove that functions like those he would call Fuchsian could not exist. Every night he went to sleep without result. One morning, while drinking coffee, the ideas came to him with a sudden certainty. The transformations he had used to define those functions were identical to those of non-Euclidean geometry. He knew it at a glance, without having completed any formal proof. Then, there was another episode. He left Caen to participate in a geological excursion. As he boarded an omnibus, the moment his foot touched the step, the idea came that the transformations he used to define Fuchsian functions were identical to those of non-Euclidean geometry. He wasn't thinking about mathematics at that moment. The conversation during the journey hadn't given him time to verify it, but he had absolute certainty. Upon returning to Caen, he verified the result, and it was correct. What Poincaré explicitly pointed out was that previous conscious work was necessary for unconscious work to occur, but the unconscious work was not reducible to the conscious. It was qualitatively different. It produced something the conscious work had not found. And Penrose considers that qualitative difference not just a psychological curiosity. It is evidence of a processing architecture that cannot be described in terms of algorithmic symbol manipulation. What Poincaré was describing, and what Penrose considers evidence of a type of processing going beyond the algorithmic, is that conscious-directed work was not sufficient to solve the problem. An interruption of directed processing was necessary, a period when the mind was not actively working on the problem, for the solution to emerge. The processing that produced the insight was not accessible to introspection during active wakefulness. It occurred somewhere other than the ordinary conscious focus. Darkness, in that context, is not just the absence of light. It is a condition favoring the type of processing active wakefulness suppresses in the face of continuous stimuli. It does not guarantee insight. It is not a meditation technique or a promise of creativity. It is simply the condition under which the nervous system can operate with less interference from the most dominant external input that exists, and in which the processes of integration, consolidation, and connection occurring in the background of cognitive activity have more room to operate. Now, I need to speak to you directly, because there is something in all this having a very simple practical consequence that is very difficult to implement in contemporary life. Visual input is constant in modern life. Screens, artificial lighting, notifications, the continuous flow of visual information characterizing every waking hour for most people in contemporary societies. That flow doesn't just occupy the visual cortex's resources. It continuously activates the sympathetic nervous system through perceptual novelty and the orienting response, suppresses melatonin synthesis through artificial lighting, and prevents the processing modes associated with alpha and theta waves from having the space they need to operate. 10 minutes of complete darkness is neither a luxury nor a spiritual practice. It is the restoration of a condition the nervous system evolved, experiencing regularly. Before artificial lighting, nocturnal darkness was a universal and unavoidable condition. The human brain has molecular mechanisms, neural circuits, and cortical activity patterns selected in an environment where darkness was a normal part of every 24-hour cycle. Those mechanisms are still present. They are still functional. They simply do not receive the signal activating them with sufficient regularity. Penrose would have added an observation that, coming from him, carries particular weight. The types of thought he values most, those producing genuine insight, those recognizing structures no algorithmic process can reach, are precisely those ordinary cognitive life makes most difficult. The continuous flow of external input, the constant demand for response and directed processing, the perpetual interruptions of attention, all favor the algorithmic processing mode machines can do faster and better. What darkness facilitates? Spontaneous processing, undirected integration, the emergence of connections directed thought failed to find, is what machines cannot do. Not because they lack sufficient computing power, but because they lack the architecture making it possible. So keep this image. There is a system in your brain functioning through millions of years of evolution, calibrated by the planet's rhythm of light and darkness, regulating when maintenance, consolidation, and integration processes activate. That system does not distinguish between nocturnal darkness and voluntary darkness. It responds to the absence of light with the same mechanisms regardless of the time of day. 10 minutes in complete darkness sends that system the signal it was designed to receive. Not as a metaphor, as photochemistry of retinal melanopsin, as an electrical signal to the suprachiasmatic nucleus, as the initiation of the molecular cascade leading to melatonin synthesis, as an increase in alpha and theta oscillations in the visual and frontal cortices, as the opening of the processing space. Constant visual input keeps continuously occupied. Darkness is not the absence of something. It is the presence of a state the brain knows, for which it has mechanisms prepared and which modern life denies it with a regularity no human ancestor could have imagined. Restoring it, even for 10 minutes, is not a poetic gesture. It is returning to the system the signal it needs to do what it knows how to do when no one is interrupting it.